Right, well, thank you for that, Andrew. Just as by way of introduction, so my name is Nigel Stark. I've lived in the village with my wife for about 40 plus years, all within Angle End. And this is about my time at what was the Plant Breeding Institute in Trumpington and its successor organisations. And I worked there for a total of about 37 years before the organisation finally sort of downsized severely and decamped from Trumpington, which is now a big housing estate. It's interesting and sad for me that there's not a single place landmark on that site now, which reminds me of what used to happen there. But anyway, I joined there in the very early 1970s when I saw a job advertised for an assistant to join a team doing the looking at the improvement of the barley crop, particularly with increasing the protein yield, particularly with regard to an amino acid called lysine. Somebody apparently a couple of years before me had been screening lots of different varieties of barley that were held in a library. These were lots of ancient barleys that were collected from around the world. And they discovered that a few of them had these very high levels of lysine and nitrogen in the grain. And wouldn't it be nice if we could actually transfer that into a more adapted crop for growing into this country? So we didn't need to substitute with feeding pigs with meat meal and fish meal and other expensive proteins, but the barley crop could do it itself if we could find a way of getting the genetics in there. Anyway, I started on this and it sounded really interesting, but it was a lab job. And for those of you who've done lab jobs, after a while, it just gets routine. And I'm afraid to say, folks, I got bored by it. But I didn't get bored by the place. It was a nice, vibrant place to work. There were about 250 people, a lot of young people there. And so I decided that I was going to look for an opportunity to get outside and join the breeding teams. And I had to wait for a while until an opportunity came up. And as an internal candidate, I was given preference. And actually, to my surprise, I got the job in the barley breeding team. And it went from there, really. I soon discovered I had a knack with plants. I liked them. It was easy to handle them. They were easy to grow. I enjoyed the work. And I got myself involved in plant breeding. By sheer accident, wasn't a deliberate career choice at all. And if I move on to the next slide, really, I'll just take you through what we did, because this was the objective, really, to improve the crops that the farmers were growing for consumption in Britain. So this was looking at varieties of wheat and barley and oats primarily to improve the quality, the yield, the disease resistance of all these things. And of course, I'd be very surprised if anybody here listening tonight, looking in tonight, can put their hand up and they say they don't have porridge for breakfast or toast or croissants or drink a pint of beer at some point. And so these are all sort of established things that go on in our civilization. So anyway, that was the objective. And of course, looking back, we've been doing this for a long time. So these are murals from ancient Egypt, actually. I think these were things that were discovered when the Egyptian tombs were opened up at the beginning of the 20th century. And surprise, surprise, people were growing wheat in Egypt 6,000 years ago. And interestingly enough, we had a collection of cereals from around the world, wheat and barley and oats. And in fact, some of them were collected from these vessels that were found in ancient Egyptian tombs. And in fact, somebody managed to get some of them to grow, astonishingly. And we were actually able to grow the very wheat that people were growing in the Nile Delta some 6,000 years ago. Very strange looking things, very long, slim, thin ears. Some of them were branched. They had bits coming out of the side. and they certainly wouldn't have yielded anything that that Chris would have wanted to have put through, grown in his field and put through his combine now. But anyway, so wheat was already known and being used, sort of, if you like, on a farming scale, and to provide lots of other people with bread. We fast forward a little bit. Meanwhile, in Britain, so this is a sort of a mock-up of an Iron Age settlement. And we know from archaeology that the early Iron Age settlers in Britain too were growing wheat. In fact, the irony was that when the old Plant Breeding Institute was finally demolished, before they built the new building site, they had to do archaeological excavation. And guess what? They found grain pits. And the grain pits there had been used for storing barley and wheat. So it's quite clear that these people 2,500, 3,000 years ago were growing wheat that was obviously suitable for human consumption. And they were using it to organize their society so they could have a stable food supply. You know, we'd moved already at this time from a hunter-gatherer society to an agrarian society. And so, yes, these people were growing and developing, and actually, probably, although they didn't know it at the time, they too were passively selecting the wheat and barley varieties that they wanted to grow for the next season. Then, of course, the Romans turned up. This was something big for us. So just over 2,000 years ago, around that, the Romans turned up on the shores of Sussex and Kent, and came to dominate Britain for nearly 400 years. But of course, that gave us access down to all the way down here into Egypt, and certainly, and the Near East here, areas around modern day Syria, Iraq, Israel, Jordan, and round into Egypt, that had been cultivating wheat also for a long, long time. And it's pretty obvious that some of those wheat varieties would have eventually found their way into Britain, I'm pretty sure. And that they would have been bringing in, I mean, the Roman army was huge. They needed to eat something when they were on the march. And I'm pretty sure that they took supplies of wheat with them. So that would have been another influence on cereal growing in Britain about 2,000 years ago. Of course, the Romans left. And then we have an odd period where we really don't know much about what happened in the Dark Ages. Very little recorded history about it, and certainly nothing about wheat production, barley production, use and growing of any crops, really. But it's pretty clear that it had carried on. Communities here would have carried on growing their cereals to make their breads and their pastries, and of course, brew their beer. Beer was quite important. And I put this slide up here. I actually found this only just a day or two ago. I was going through my records, and this was a presentation that I found that was made into PBI in 1987, looking at the progression of the development of wheat. And so if you start on the left there, that funny little spindly thing, they are actually ancient predecessors of wheat. And you can see the progress of the different types as we go through. And actually a modern wheat, which Chris would recognize up on the top right-hand side there of that slide, is pretty much what they look like now, but that's a much more complicated crop. For those of you who know a bit about genetics, modern wheat is actually a hexaploid. So we as humans are diploid. In other words, we have 2 copies of the genes in us, but actually in wheat, there are 6. So it's quite advanced. How it quite got there, we don't actually know. There's no evidence that people actually did it deliberately, but at some point, these simple ancient diploid crops, and at some point, this is like a durum wheat here, where I've got my cursor going up, and the durum wheat is actually tetraploid. That's 4 copies of the genes in there. I don't want to get this to be too technical at this stage, but just to show you how it's developed over this time into quite a complex crop from very, very simple origins. And indeed, when I was at PBI, we used these very ancient varieties to do some crossing and hybridization to introduce some of the interesting characters that exist in them for instance, disease resistance, to transfer them into modern crops. So if I look at the next one, where do we go? Yes, so of course, during the Middle Ages, we were doing strip farming. Tilling was very simple. Everything was drawn by horses or oxen to plough, harrow the fields, but there was no mechanical harvesting at all. And so medieval living was done in small villages where people lived fairly close together, and all their strips were around the edge, and pretty much bread was a staple variety. Don't forget, folks, that at this point, potatoes had not yet been discovered. They were something that existed in South America. Okay, Francis Drake brought them back in the early days from his travels. And there's that famous picture of him presenting potatoes to Queen Elizabeth, who was on the throne at the time. But it took a long, long time for things like potatoes to come into our general diet in this country. So yeah, bread was still fairly staple. You would have had it most days. I wonder how many of you have seen this. This is a painting by Bruegel, Peter Bruegel, the elder, Dutch artist in the mid 1500s. And yeah, that's probably pretty accurate. Those people aren't dwarves. Okay, they're probably shorter than we are on average height today. But yeah, wheat was pretty big stuff, pretty tall. It all had to be harvested by hand. And if you think back to that slide I showed you of an Iron Age settlement, there's an interesting thing about that, another use of wheat, which of course was its straw. And of course the straw was used for thatching, and it kept people dry. It was really important for shelter. So nothing of this would have been wasted at all. The ears would have been tiny little things on, as you can see, on the top of these great big long straws, representing probably at a maximum 15 to 20% of the overall weight of the crop. So lots of straw, tiny little ears on the top. And if you're lucky, Chris will know this, in a good year, you might have got 1 ton of seed from every acre that was sown. And of course, the people sitting around the tree, some of them are drunk, so they're having their small beer, which had been brewed from barley at the time. Similarly, would have been very, very tall, very floppy, small ears on the top, but barley straw would have been much more of use for things like bedding of animals. Right, why has that gone blank? Ah, yes, here we go. So this is where it all starts. So this is Gregory Mendel. He was a Moravian monk who worked at a monastery outside the modern day city of Brno, which is in Czech Republic. And he submitted his work on, now what's it, the German, well, the cultivation of, yeah, where are we? Okay, yes, so I've got the German translation here. So he produced the laws of genetic inheritance, and he submitted that work. He worked on this for about 12 years apparently, and submitted his work, which was written in German. And I think the Czech Republic was probably part of the Austro-Hungarian Empire at the time, but a copy was submitted to the University of Cambridge. And I'm afraid he got overshadowed by someone who came just before him. And of course, you know who that is, Charles Darwin. Of course, Charlie Darwin created a huge rumpus, didn't he? His theory of evolution. And Gregor Mendel's work was basically forgotten. But he did some very interesting early experiments. I mean, he didn't know, for example, the existence of DNA at all. He didn't know about that, or genes. But his work inferred it. And what he did actually, he's worked with sweet peas. And you can see him standing in front of his crop of sweet peas here. And what he did was he hybridized groups of white and purple flowered sweet peas, and found that the results, when you crossed a white flowered pea with a purple flowered pea, that the resulting next generation of peas always separated out into a 3-to-1 ratio. 3 of them were purple and only 1 was white, however many times he did this. And so he deduced that, of course, that the genes in the purple flower were stronger, much stronger than the genes in the white flower. He didn't know why, and he also did the same thing with wrinkled peas and smooth round peas. And he found that the round pea was always the dominant type to the wrinkled pea. And he suggested therefore, that this gave an avenue to be able to people to look at cultivated plants and hybridize them, and gave them a method for selecting out types. And interestingly, some of the characters that we look for today indeed are so-called recessive genes. In other words, they appear much less frequently, and you only get that combination of white when you have both of the genes that confer the white color actually join together in the cell. If you get a white gene that goes for white color and a gene for purple color, it's the purple color that wins out until you get a pair of genes that are white. So anyway, his work lay in the University of Cambridge for jolly nearly 40 years. And it wasn't until this chap suddenly the penny dropped with him and a group of others about the potential for this. And it's quite an amusing slide, because you see him with this huge mock-up of a wheat ear, and he saw the potential for using Mendel's techniques to cross cereal varieties, particularly wheat, of course, he was interested in. And don't forget, at that time, Britain's population had exploded in the Victorian era. It had gone up by 4 times in 100 years. We were importing huge amounts of wheat for British bakeries from Canada, huge amounts. And he was saying, well, you know, if we can use the genetic techniques as explained by Mendel, we can do it here. And in fact, he was very successful, and working from the University Department of Botany, he very soon developed a variety called Little Joss, and as inferred by its name, it was shorter than most other wheats at the time, much less straw, but had much bigger ears, and the yield of grain was significantly larger. And so much were the bosses at Cambridge University impressed that they set up the Department of Plant Breeding in 1912, that was. And so with the variety Little Joss, which was soon multiplied up and sent out to farmers to get into general cultivation, soon after that came another variety of wheat called Yeoman, a variety of barley called Sprat Archer, which was, believe it or not, a cross between a variety called Sprat and one called Archer. By the way, these names were given to land races. And land races were varieties that have been grown for years and years and years. And in different parts of the country, there were different races. So in the southwest of England, there were varieties called Wiltshire Common. There was a variety called the Reverend Chevalier, which implied that a man of the cloth had actually gone out and selected larger ears from a field and found some and grown them on and multiplied them. And anyway, the Department of Plant Breeding was formed in 1912 and became quite successful. And it worked out of fields up at Huntington Road on the university farm there. And then in 1948, after the Second World War, the government decided that we really needed to get agricultural research a real boost. Shortages of food in the war, and don't forget 1948, there was a lot of rationing going on, and it was decided that the government should set up a whole host of different research institutes all over the country, from Scotland, Northern Ireland, Wales, right down into the south of England, each to specialize in a different aspect of agriculture. And PBI was one of those. There were ones at Houghton, there was a poultry breeding research, there was the Institute of Animal Physiology not far away at Baberham, the Rothamsted Research Council down at Harpenden in Herts, John Innes Institute at Norwich. These were all part of that same group. And but it needed to expand, and so it came out of the university and was established at Trumpington, just off Maris Lane. And these buildings were built during the early 50s, and it was finally occupied in 1954, when I was very small, actually. And it started from there. And PBI was then sort of, if you like, a standalone entity away from Cambridge University. And there it is. So that's a picture of the building. That was the main building as it was, and very much as I remember it when I joined in the 1970s. It was quite a big complex, in all about 11, 12 acres of office buildings at the front there. Behind there were all huge complex of laboratories, glass houses, workshops, stores. And in the end, by the time I joined, there were about 250 people. It was quite a buzzing place. All aspects of arable crops were there, so not just cereals. There were beans, peas, potatoes, oilseeds, grasses, clovers, all that kind of thing. And during the 1970s and early 80s, it became very successful. There was something called the Queen's Award for Industry at the time, and it was awarded to scientific, technical innovation to various companies throughout Britain. And we won this for a variety of things, our successes in plant breeding. Here we are, some of the notable varieties. Some of you might recognize these names. Maris Piper potato was bred there in 1966. And of course, why wouldn't you grow a potato that was developed in Trumpington if you had an allotment 200 yards away? It was the obvious choice. And of course, Maris Piper potatoes we still grow today. You can still see them advertised. You can still buy them in supermarkets and go into a chip shop and they'll tell you, the new season's Piper. Maris Otter, still very much loved by the Real Ale Brewing Club. It's astonishingly, it's still the benchmark for malting barley. If you want the optimum type of malting barley, you go back to this variety, bred, well, not just bred, it was released in 1965 to the farming market, the agricultural market. So it was developed during the 1950s. It has an appalling history now. I mean, it drops dead with all sorts of diseases, and to keep it going, you need to spray it with all kinds of fungicides. And the yield is very, very much smaller than you would accept from a modern crop. But it commands a very nice premium. And if you want premium ale, real ale, a lot of these small brewing outfits will go out of their way to find a source of Maris Otter. Other things like Maris Huntsman was the first wheat to break the 4 tons per acre barrier. Remember that I told you about that the yield would be about 1 ton an acre if you were lucky back in the medieval times. So that had quadrupled by the time we got to Maris Huntsman. So that was another milestone, and that was one of the things we won the Queen's Award for Industry for. It wasn't a particularly attractive wheat. It wasn't particularly good at making bread, and the grain was rather thin, but boy, were there lots of them. And then on this last thing, Hobbit, this has got nothing to do with Tolkien. He didn't work with us at all, but Maris Hobbit was the first of a generation of dwarf or semi-dwarf wheats. And of course, the demand for straw had slumped. There was no need to thatch roofs. The number of thatched roofs in the country had probably gone down to probably less than 1% by that time of the overall housing stock, maybe even smaller. Nobody really wanted the straw. It was a bit of an embarrassment. Farmers at the time were often cutting it with the combine, and then, I don't know if you remember, we used to have these fires in the field. People just used to burn the straw to get rid of it. It wasn't worth very much. And so what we did at the time was use a gene from a small wild type wheat, which was actually found in Mexico. And we were able to transfer a lot of the genetic information from our adapted types then into this dwarf wheat. And interestingly, what we had done is we hadn't changed since the Middle Ages. The overall yield of plant biomass from above the ground was exactly the same, except that if you remember that picture I showed you for Bruegel, the grain yield would have probably been around 10, 15, 20% at the most in a good year. When we look at the modern wheats, the grain at the top represents more than 50% of the total weight of the plant. So what we've done effectively is we've made them shorter, but actually use the energy of the plant to divert its resources into growing grain rather than straw. So that was one of our achievements. And of course, semi-dwarf wheats nowadays are pretty much what all farmers would be growing right through till now. So here we go. So let's getting on to what happens in a breeding program. These are our objectives. So first and foremost, Chris would understand this, grain yield. If it doesn't yield well, that's what you get paid for on the farm. That's where your money comes from. So that's a primary importance. Of course, disease resistance, that's really important. If the poor plant gets covered in disease, very often the first indication is that the yield is affected, the grain is often shrunken, the plant itself may be shrunken. Standing power, that's important. It has to stand up to summer storms. There's no good growing your wheat variety and then on the first big storm in August. And if you remember last year, yeah, August was pretty wet, but I didn't see very many wheat crops go down last year, but it's important. And again, resistance to sprouting. If you get a very, very wet season, the grain will actually grow in the ear. And I've seen it in some seasons where you get this green haze coming out of the top of an unharvested wheat crop. And of course they've sprouted. And by the time they get to that stage, it's virtually worthless. It's not worth it. It certainly won't make you bread or biscuits or pastry. It might just be worth harvesting to get it off the field, dry it, and feed it to animals. Coming down the list, quality traits are really important. Certainly bakers would be interested in how good the baking quality is. This would all be down to how well nowadays the bread rises. We all expect this nice big spongy loaf with plenty of air mass in it, a nice sort of open structure inside. We don't want it to be thick and heavy with a soggy bottom, as Mary Berry might say. And there are all sorts of parameters here. Would it make good Weetabix or Shreddies? And for barley, of course, would it make good beer, or would it have the right characteristics to make a good malt whiskey, for example? And then one I've put in here at the bottom, coming up the agenda very much now on plant breeders list with these long dry periods, is drought tolerance. Really quite important, you know, and again, that's down to the ability of the plant's roots to get down a long way. And the root of a wheat will actually go down over 6 meters. We did these experiments back in the 1970s. I remember some experiments out in the field where we traced the roots of the wheat plants, and they went down nearly 20 feet. So they can be quite deep, but of course, you've got to have the soil structure to allow them to get there. Okay, I didn't want to get too technical, but this is a breeding scheme, and this just shows you the number of lines that we'd be. So each line here represents effectively a growing season or a year. So it takes 11 years to develop a crop, if all goes well, from the time when you first make your hybrid between your two parents until you get something which you think has got an advantage to be able to send it to multiply it up and market it for farmers to grow. So if I take you briefly through this, an F1, that's the first cross. That's the hybrid between your choice of male and female plants And they would have the characteristics, obviously, that you'd want to combine to have the best of the possible new variety that you're looking for. It doesn't always work. Sometimes you can have two really good varieties and you cross them and you realize that the resulting generation is also ran, that you've got the best possible combination in your parents, and when you cross them, somehow that's lost. So some varieties make really good parents and others don't. So anyway, in your first generation, your F1, what happens is you get something called hybrid vigor or heterosis, and all those F1s are the same, and they all show the dominant characteristics of those two parents. And very often that yield is somewhere around 25, possibly 30% higher in that generation. And that's why when you buy your vegetable seeds, your F1 crops are always the ones that yield better. But of course, when you grow them again, don't forget these are self-pollinating crops. Wheat and barley and oats are all self-pollinated. So by the time you get to F2, what's happening here? All those genes from those two parents, pretty much at random, although of course, that's the rub. It's not always at random, but we don't know how random it is until we've gone through the series. All those plants are recombining into all sorts of different possibilities. So for instance, if you've got parent A, which is really tall, but has fantastic grain, but falls over, you want to cross it with parent B, which is very much shorter, not such good grain, but has terrific disease resistance. So your objective is to combine the plant, try and get a plant with really good disease resistance and big bold grain that doesn't fall over. But of course, when you recombine all those genes, you get the other things. You get the big ones with tiny grain, with no disease resistance that fall over. So you really don't know what you're getting. And we're looking at 1 million plants. So there's 1 million plants in the field there. Sometimes you'll go through these plants and you won't take anything from across because you think there's nothing interesting. And other times you think, wow, some really interesting things. And this is very much hand labor. So you'd go around and you would put a tag on all of the plants that you're interested in. You actually go around the field, walk around the field and physically hang a label of some of the plants you think you want from that. And out of those 1 million plants, you might choose just 50,000. So most of them are rubbish, utter rubbish, but you don't know which ones are rubbish until you've gone through and looked at them. More segregation takes place after that, so you've got a lot of more recombining, and the next generation, the genes are beginning now to line up more, so you've got some of the same characteristics on both sets of genes. So you might choose, it's got 80,000 here. I think that's a mistake on this, I've just realized. It would be less than that. It would be somewhere between probably 10 and 12,000. So continuously, as I go down through this line here, you're eliminating things. So 4,780. And by the time you get to this sort of 80 and 10, these 10 are beginning to look really interesting. Got all the characteristics. This time, you're beginning to trial them. This NL thing implies that NL is a national list. It's a list which is issued by the National Institute of Agricultural Botany every year, which shows farmers which are the best varieties to grow, what their characteristics are, and gives them an idea of what to choose when they're buying seed. So here we go. So by the time we get to F8, this is the point at which we're actually sending quite large quantities of seed, 50K, 100K units out to be trialed up and down the country. And we would also be doing our own testing at the same time. And with any luck, by the time we get down to year 11, this is RL stands for recommended list. If you're lucky, after about 11 years, you can go from your first hybrid you've made in the glasshouse down to something here, which is equal to, or if not better than anything else that's around. So here we are. It's all very much manual labor. Here's one of my colleagues, Hilary. She's actually doing some crossing here. So the way you do this, because they're self-fertile, so this is normally done in a glass house, you've got controlled conditions. So what you do is when the plant grows up, you get a point at which you have to go and emasculate the plant. Did I see anybody twitch then? So what you're doing there is you cut open the florets, you trim them back, and you pick out the immature anther. And you've got 3 anthers per floret. You've got, depending on the size of the ear, you've got in the region of 30 to 40 florets on a single spike. And you, on the wheat plant, you prise them open, you pick off the outside bits, which is the lemmas, and you go in and you pick 3 immature anthers from each of those florets. And then you stick a bag over the top because you don't want anything else to get in there. And after about 2 or 3 days, the whole thing matures and the stigma in that plant, which is the female part, will be maturing. And what you then do is choose which male you're going to add, which cross you're going to make, and you'll have those all lined up on a bench in the greenhouse, and you'll go and take pollen, which is maturing. And you know that because when you open those florets up, when you cut them back, you add a bit of heat and light, and all of a sudden they dry and expand. The process is called dehiscent. So when they're dehiscent, they're ready just to spill all those millions of grains of pollen out. And what you do is you work up the ear as quickly as you can, getting that pollen, which is just at the right point, onto the top of a stigma, brush it on so that the pollen tube will grow down into the ovary and fertilize that plant. You put the bag over the top, and that's your next, that is the beginning of your next generation. And say on an average season, a program would make about 700 of those. It's quite intense, quite labor intensive. You don't get weekends off when plants are growing like this. You just keep on coming in and doing what you can. The whole process, if you spread the sowings out, probably takes about 3 to 4 weeks. Very intense, very labor intensive, highly skilled. You have to get to know just when the plant is ready, but it works. And nowadays we speed the process up by using controlled environment chambers. DNA technology enables us to check which particular, we have things called markers. So these are genes we know which exist in here. So if we're looking, for instance, for a particular marker for bread making wheat, and we're making a cross for bread making wheats, we will grow the next generation of F2s in trays here, and we'll take a leaf sample off each one of these, snip it, put it into the test tube, extract the DNA, and then I haven't got a picture here, but it's all done by robot. These robots come along and take samples out and process them, and that will tell the breeder which of those individual plants there are worth keeping and which can be discarded. And that saves you a whole generation of going through the field and harvesting and putting it through the lab. And so you can do an early stage selection for particular genes at that point. Yep, so these are some of the common diseases we look at. On the left here, we've got mildew. This is a barley. Actually, this is a 6-row barley. But as you can see, the effect of disease can be quite significant. These white mycelium, which is growing all over here, over the top, can affect the yield hugely, can reduce yields by up to 50% if it gets out of hand. And similarly on this one here, this is a barley plant. This is a disease called, I think this is Rhynchosporium. These little patches down here on the leaves, it destroys the photosynthetic area. And of course, when you've got a huge field of this, if all the variety is the same, then the disease, pretty much like a coronavirus, if everybody's susceptible, it goes rampant through this crop and can take all these out in a very, very short time. So this is the point where you can control these diseases using pesticides, but it's best to start with a plant that has some level of resistance in the first place. When we get to a later stage, these are field plots. You can see the M11 in the distance here. So this is the site at down at Ickleton, out of the village there, towards Elmdon, which is where the rump of the successor organisation from PBI now does its wheat breeding, and you can see here this is quite a nice drone footage showing the subtle colour differences within each one of these wheat crops here. So each one of those plots is about 6 meters long and about 1.5 meters wide, and they will all be harvested individually. But during this stage here, it would be the job of the breeder to work through each one of those plots several times during a growing season to look for all the aspects we're looking at. If they've got disease, how well they stand, how good looking the ears are. You just get a feel for whether it's going to produce enough stems and its general standing ability. So we'll be making observations on those throughout the growing season. And that's another one. Actually, that's taken from that's footage from an ex-colleague in France in the field. Again, showing these huge colour differences. And you can see these will be yield trials. So they're all sown as a block, but then just before harvest, these long straights here, or we call them tram lines, will be taken out with a tiny little narrow combine with a cut of only about 5 foot wide. Will go through here, take all these out that way, and then each plot will be harvested individually here into a bag, so we can assess its yield. So we weigh the grain, and then later on, of course, the grain we will be using for things like baking or other quality tests that we would do. So yeah, there's a field operation. That's pretty much what it looks like. So here we go. You see these little blue mini combines here. They've got all the same moving parts as the big commercial ones, but they're designed for taking small samples, just a 5 foot cut on the header, on the blades here. But you have someone standing at the back here, bagging off the seed from each plot to be taken away on these trailers and back to the laboratories to be weighed and to be sampled and stored, and we decide where they go from there. So yeah, a very, very labor intensive organization, as you see, lots of people. Very often it's without temporary workers at harvest, the operation wouldn't exist. So, and there we are. And of course, we also develop larger scale. So when we think we've got things that are going to make commercial varieties, we're harvesting with larger combines. This is still tiny compared with what the modern day farmers would have, but this is just right. This is somewhere about probably a 10, 12 foot cut on this John Deere combine, because we'd be taking somewhere in the region of about, somewhere in the region of about 3 to 5 tons from crops at this stage. And then they would go for much bigger scale tests for bread making or tests for malting and microbrewing. We can speed the process up. So speeding the process up for convention, other than conventional ways, chemical mutation, this was used in the past. In fact, there was a famous variety of barley called Golden Promise, which is still used, which is very good malting and brewing characteristics. It's still used in some part. It grows particularly well in the Scottish environment, and that was developed by putting seeds into a solution of sodium azide for 48 hours, which of course is a well-known chemical mutant. And you take them out and you dry them, and of course most of them are killed by the process, but those that do survive, you've got chemical mutations. So you've got an instant sort of, if you like, way of shifting the genes about without having to go through the process. Another one is called double haploid method. And this way you actually can cross a barley or a wheat, and you can transfer maize pollen or pollen from a very ancient barley called Hordeum bulbosum. And what happens is that the plant initially accepts it. So the pollen, so it gets fertilized, the pollen tube grows down, the embryo gets fertilized, but then it realizes something's not quite right. And just at that stage, you put a chromosome multiplying agent, and colchicine has the same effect. And you put colchicine on it, so it immediately doubles the number of chromosomes you've got. But of course, they're both true sets. It's an identical copy. So you can look at these things instantly and see what your genes are, and you know that's a pure line. Single seed descent, that's another method we've used. So this is growing plants in very, very small cells, about an inch square, and you force them into producing flowers really, really quickly. Traditionally, you would say out in the field, if you've got a conventional program, you would sow a wheat variety in October, and you wouldn't harvest it until the following August. That's one season. With single seed descent, you can do the same thing in 14 weeks. You can grow it, you can give it that minimum period of short days, cold temperatures, bring it out, give it high temperatures, lots of light, and all of a sudden it shoots up, gives you a single stem, probably no more than about a foot high or so, with a very tiny little ear containing perhaps less than 10 grains, and you pick one grain off and you sow that again the following season. And that way you accelerate the breeding process. And then last of all, you can use out of season nurseries. And this is where I first got to go to New Zealand as a plant breeder back in the early 80s, because we could grow spring barley here, sow it in March, harvest it in August. We could turn it around really quickly, so we could thrash it, put it into packets, label it, and we'd put it on a plane, send it over to our sister organisation at Massey University in Palmerston North in New Zealand, and they would then grow it in their fields. So they would sow it in middle of October, and then it was one of our jobs to go out and then look at this nursery from January through to harvest in something like about late February, early March. It was our job to select the lines out there, process the plants, and send the seed back just in time for another sowing season back here at Cambridge. So by sending it the other side of the world, again, you could double it up. But the advantage of that is, of course, you've got two natural growing seasons. You can use single seed descent, but of course you're growing it quickly under artificial conditions. Here, the climate pretty much, now where's Palmerston? It's down, Palmerston's, no, here it is. That's Palmerston where my arrow is there. So the conditions there are pretty similar to what we'd experience here in southern Britain. Day length slightly different because of course they are 40 degrees south, whereas we are 52 degrees north. So their days aren't quite as long in the summer and their days aren't quite as short in the winter. But for a spring crop, that doesn't really matter. And that works, and that's still going on. My former colleagues still traipse forward, backwards and forwards to New Zealand every year, sending their crops out there. And then finally, back in, it was only a couple of years ago, finally scientists have completely finished the sequencing of the wheat genome. I think this was done for the human genome in about 2010. Hilary, you might know more about this than I do, but this was finally done. So we identified about 96,000 genes. So it's just to show the sheer, this slide really, just to show the sheer complexity of what the wheat genome has. Huge number of things to grow there. And that's why the process is not easy to automate. And that's why it's still plenty of scope for individual breeding companies to apply their own art, as it is still as much an art as it is to a science. And of course, the fascinating thing is all those genes are in there. And what makes plant breeding so interesting and sometimes so frustrating is that sometimes you've got very different genes for different characters so close together that they never split. They always transfer together. And of course, this is where GM comes in, and you can do it. But by traditional breeding methods, you've always got, for example, I know in the potato crop, blight resistance is very, very closely associated with cooking quality. And plant potato breeders for decades have been trying to separate the two out with natural means and selection. And it still is almost impossible to get that combination where you get a blight-resistant potato that produces wonderful cooking characteristics. So there we are, folks.